Welcome, everyone. This is really the special webinar event. It's special because we normally invite people who are in academia, but today we have invited a very experienced researcher and entrepreneur who's in the area of AI-enabled drug discovery. I also think that many of you may have already known that I've started a new center called the Systems Pharmacology AI Research Center. And this is indeed the first webinar, just maybe in the name of the center. And so it's very proper that we position the first webinar in both academia and the industry, a slightly different industry than healthcare, and which is the pharmaceutical industry. So let me introduce Dr. Lurong Pan. Dr. Lurong Pan has a bachelor degree in applied chemistry from Nanjing University in China, a very good university. And then she got her PhD from UAB in chemistry. She also got a Master of Computer Science from Georgia Tech. In her journey as a computational chemist who have found this drug discovery her passion, she has garnished more than 10 years of research and development experience applying computational techniques to study disease biology and study drug design. She has started her own company, is a founder, a CEO, and also she has been affiliated with UAB, I believe in the School of Engineering, to help with computational sciences. We're very happy that she can share some of her experience, and both very practical in the industry, and also her research in her company gave us some insight on what her view of the future of medicine and AI will combine. So without further ado, let's welcome.
Yeah, thank you, Jack. So everybody can also hear me from the Zoom? Okay.
Yes.
All right, thank you. Yeah, today I'm going to give a relatively broad topic on how artificial intelligence being applied in the pharmaceutical industry and some of the current progress and the future challenges and the methodologies. So I try to concentrate on all that in like about 40 minutes, and then another 20 minutes we can have a free discussion. So I will first introduce myself and how we are how myself being brought into the field, and what the research and business venture have been done in this process. And then I will briefly introduce about what is drug discovery. Probably all of us are working in the huge system, but it's just on one part of it. And right now I'm just going to introduce the whole picture and how AI applied in different steps in the drug discovery process, and why we need AI and how the AI method being effective in the process. And then I will talk about some of my company's business, about how we commercially apply certain technology in the industry. So yeah, a little bit about the overview. So I, like Dr. Chen already introduced, so I have a long years of education. I've been basically study chemistry and computer science together, and most of my research has been done in the computational biology space. And I had a brief industry experience in artificial intelligence from a company in Boston, and also I have a project overseas with the Gates Foundation that to establish an internal AI drug discovery platform. And then in 2021, I started my own software company that leverage certain AI model to do drug design. So I will talk about those journey briefly before I go into the detail. So my PhD in UAB was quite happy that so we started very fundamental how to use quantum mechanics to simulate proteins and study disease biology. So that was about 5 years research, but let me learned all the methodology from the very commonly used method in the drug discoveries, quantum mechanics and molecular dynamics. And then we can use them to study very precisely on molecular interactions, and also in a bigger picture, the disease biology process we cannot observe from microscope, but can be simulated. And then I continued my postdoc research on larger system, from a small peptide to a transmembrane protein, and also some of the membrane proteins assemblies that with Dr. Eller and Dr. Sigrist. So those are, at that time, the largest scale of biologic molecule simulation in the world, that we leverage the entire like NSF, we call it exit clusters across multiple campus to simulate only like about 1 microsecond. And this is by far the limitation of this methodology, is this how far and how big we can simulate. So at the time, I feel like in order for us to really understand the biology process in a more precise and larger scale in vivo way, this methodology has to come to its like final limitation, is just basically a single protein, or at a scale of like a membrane or a single, you know, like actually by far, I think the biggest system was just a single virus that this method can simulate, and we can't do anything bigger than that. So then I go to at the time, I was working with a bunch of scientists, engineering department. We tried to basically to solve the simulation problem that we call the multi-scale efficiency algorithm, to find an algorithm that we can simulate not just at the physics or chemistry level, also biology level. Then we will have to use more smart approach. And machine learning and deep learning is one of it. And we also invented other algorithms as well. And this center is also promoting collaboration between like industry, and also like medical research, and also business people. So it's like a very vibrant, very open-ended place in engineering school that we've been collaborating and have brainstorming a lot of idea, and a lot of company has been formed since 2015. We started the center. And then at the time, I also take another degree from Georgia Tech, dive deeply on how machine learning could apply in medical science. And one of the projects that I did was basically predict the patient mortality in the ICU, leverage all the medical history of a person from all the image data or electronic health records. All the medicine is being taken, and all the vital signs currently monitored. And I invented a dynamic observing window, which we basically has to dynamically identify or set that parameter of how long the history of a person's medical history has to be learned, and to predict and how length of the window, and be able to achieve a pretty good accuracy. That I started a company primarily in California, because there's a lot of entrepreneur like people that kind of really like this type of idea. And so basically I started basically integrate some of the data I think may be useful for prediction. There's like about 30 domains we can predict to design and predict the property of a drug. And it can be very quickly done. In the past, when I do a virtual screening, it takes a graduate student probably 3 months to finish the work. And this system can only be done like automatically in 1 week. So this is kind of the merit of the software. And we can offer design of protein drugs such as antibody and small molecule PROTAC, and also like RNA sequence design. And we get some funding from NVIDIA and AWS, and also we get buyers from some certain pharmaceutical company and industry. So, and this is, so when I was a graduate student, I would have never thought about any computational methods can be commercialized, because I thought it was very, basic research. But now I think this is gonna be like a good trend in the industry. And also I was interviewed by Wall Street Journal for the company and did how it's been, we're using the same algorithm as ChatGPT in design molecule. And this is a new trend that people realize that protein and DNA sequence has no difference than human language that can be learned and interpreted in a different way. So this is myself. And now let's talk about science. And so I might get more technical from now on. So first of all, what is the drug discovery process? So this is like we call the 4D map released by NIH, which is actually the gold standards of how a drug is discovered from the beginning to the end, and which you can see probably the letters are quite small, but I would just basically just give some example. Basically, we start with translational medical research, which most UAB has been done like really great, in the world, that we discover important genes, therapeutic targets correlate to a disease. And then the industry normally would take the targets and to do all kind of target validation, and then started to like high throughput screening. And then after that, they identify a lead compound, and then they started to involve all kind of chemists and biologists and to do lead optimization and test in vitro and in vivo. And after that, they're gonna go to like we call the drug development stage, which they take the active component and to do formulation and to do the scale up chemistry, try to see if it can be mass produced with relatively low costs and with the least toxicity that the byproduct contains. So that would normally take a long time, and then we will go to clinical trial. And we have the phase 1 mostly assess the safety, and phase 2 and phase 3 for a small or large population of efficacy study, and they will take another probably 5 to 10 years. So the entire process is really a system engineering, if we can say that way. It connects both all kind of research entities and people with different expertise, and with scientists, engineering, and people in the regulatory space to really collaborate and has this common knowledge to push the one goal together. So in that case, how do we really efficiently push the process? That's really a challenge in the industry, because on average, actually 75% of the clinical trial has been failed. So, and that's average on the cost of every single drug that has been pushed in the market. Their cost about average $1 billion and 15 years conventionally. However, there are over 10,000 diseases that has no cure yet. But we already identify them. This disease exists. So this is basically the ultimate problem we would like to solve, is the huge unmet medical needs as respect to the lengthy and cost process of drug discovery. And this is the number of compound will be synthesized and tested, and eventually go through all the process and to be successfully marched. So this is just like the reality we are facing, and hopefully AI can do some good. So for those of you who has not taken any artificial intelligence class, what is AI is not hard to understand, because everybody probably already use AI one way or another. And AI could actually, in my book, AI basically can do everything human do, and sometimes it can do something human cannot do, because it can basically do something 100 human collectively do with just one machine or one algorithm. So that's the difference. And right now, most of the AI model that already developed, commercialized, already passed the Turing test, which is an expert cannot differentiate if that's done by AI or human. So that's how evolving these spaces. And we really would like AI to use in a good place. That's why actually the company name, Ainnocence, of my company it's actually stand for innocent AI, because I can totally see the potential of the AI does the harm to human race, but hopefully we can use AI in medicine that does something good to the world. And here, why we use AI, so we combat several problems in computational drug discovery. So first one is scalability problem. So first, because like I said, the conventional drug design methods has to its limit to simulate a single drug and protein interaction, which is really far from the in vivo efficacy that is a system biology of scale. So in that case, in order to really describe what's going on in a larger scale, we mostly use informatics, or we call traditional biostatistics methods to quantifiably explain what's going on in our human body and try to relate the genotype to the phenotype. And that's what it's basically. This is all the methods that are in the field. It's here, but the problem is, for example, we wanted to know precisely how one drug, which is atomistic scale of things, work on a body which is consists of 30 trillion cells, and each cell has 22 millions of proteins. That's almost impossible to simulate that grand of system, no matter how big our computer is. So in that case, we would really need a good algorithm to do it. And then for all the algorithm, there's two type of how we describe the biological system. One is we call the geometric. Basically, we just take the 3D like visualization of how the atom or molecule look like, and then to simulate it. This is like the old methods. And another is semantic, which is we just take information. It could be very abstract as an English letter, or it could be just some random numbers, and to kind of encoding our biological system. And then in that case, we based on those data, then we do the simulations and performance. So, and the second one is translational gap. So before we have AI, we have to simulate different scale of precision, use completely different physics model and assumptions and approximations. So we can simulate from a single molecule, from using electronic structure to all the way to organ. We use finite element, but the problem is each one of those models cannot translate between each other, and you can't addictive them to kind of zoom in and zoom out at using those different models. It just cannot be done. So we really require a uniformly applicable model, and deep learning is one of the models that it can leverage data from different scales and to perform like actually inference across different scales. And also another thing is another reason we use AI is to reduce computational complexity. So like for example, in the past, when we try to compute using QM method to compute a property of one molecule on one GPU, it takes a few hours. And if we are using some other high level of more approximated model, it's still gonna take a few minutes or to a day to compute that. But if you use a trained AI model, black box, to do the same prediction, like 1 minute in one CPU, we can do thousands of output. So in that case, this is really reduced the computational complexity in about 10,000 times. So then next, let's talk about how we construct the AI system. So for any given AI system, there's 3 elements is necessary: is algorithm, big data, and computing power. And then when we design the algorithm I think is the key. So we should consider how can we leverage the best of what available there for the big data, and what will be the most efficient algorithm that you use the least data but have the most accurate result, and also without sacrificing actual computing power consumption. So that's how that's how when we design algorithm, our goal is, at least in industry, but I think for academic, you have a more open-ended, invention on that end. But for machine learning algorithm, there is really commonly used algorithm, just 3. Supervised learning means we have the answer to a set of questions, we have data, just learn those data and to make sure in the future it can answer that question. And unsupervised learning, almost like an open-ended question, it lets the AI learn itself and to abstract knowledge from it without asking any question. And the reinforcement learning is giving like an environment of a force to kind of force the AI to only focus on certain aspect of a problem. So it would be more problem specific. So those are really commonly used for example, protein structure prediction, function prediction, genome engineering, system biology, and polygenomic inference. So all kind of possible problems you answer in the drug discovery chain. And there's a lot of algorithms. They are all open source available. So I think this community is quite collaborative internationally. And then there's different type of biologic problems in terms of translation from diagnose, target identification, and drug design. There's different regression, classification, clustering methods using all kind of algorithms that can help with the detailed problem. So in the future, we will envision like really we can do a precision medicine on everybody. Is basically an AI would perceive a genomic information of a person and give some kind of personalized diagnosis. And after that, they will automatically trigger a drug design engine to either search available drug or if it's unavailable, how do we design what type of therapies could precisely target this diagnosis, and then we can solve the problem very very automatically in a way. And the second the element is big data. So this is actually not. I found out when I go to industry, especially in the AI field, I feel like most of how we integrate data is not being taught in academia. And I remember at the time in 2015, the UAB has an initiative across different departments, and people have troubles, just even don't know how to store and sharing and or extract information across departments. So in that case, like we say, this drug discovery system chain is very long, from clinical data to pharmacology data. It is just very and they're spread in different institutes and in different websites. So how can we integrate them together? That's an engineering problem. And then there's a set of protocols for data QC and also the data standard and schema discovery. So those are just some of the example that I gave here. And then the last thing is computing power. So right now the computing power is almost like a new electricity. It's just everybody has to do this type of business. And the extreme, some of the very useful resources, one is Exceed. They almost have unlimited renewable computational resource to academia, and those resources normally actually it's overproduced and less utilized. So I highly recommend users to apply for it, and it's very easy to gather grants. And they normally have the SOTA, the state and the GPU available. And also in this field, Dijon Research even have their own ASAC chips being developed just to simulate molecules. It's special chip, and then he donated this to the NSF as well. So those are good resources we can use. I think for the time being, I might not dive deep into some of those challenges because mostly are very chemistry deep knowledge, but basically there are some challenges like how we use AI to describe chemistry and how to design drug and synthesize drug efficiently. But some of those challenges has already been overcome. But just I will just briefly mention some of the interesting points. So first of all, those two graph actually shows the same set of chemicals, but it depends on which dimension we look at them. They can be very similar or very different. So because we can describe a molecule from a 0D, like the molecular weight, or 1D, 2D, 3D, or 4D. and even higher, like 1,000 dimensions with AI. But human can normally only perceive like a two-dimensional, like how the molecule draws in a whiteboard. But the AI can see more dimensions, and that's why when we see that's why kind of AI and medicinal chemists can really do complementary work on how to define if two chemicals are similar or not, if they're drug-like or not. They will have very different but very complementary view on this. And sometimes in chemistry, if we change one atom, it would completely shift the property of a medicine, and those also can be solved by AI. And another thing is, when we optimize a drug, there's normally over 20 different properties we need to optimize at the same time. But in the past, expert or any human being can only optimize one at a time by some kind of linear logic. But AI can do a multi-objective optimization and find the sweet spot much quicker. And then also AI can do different type of phenotypic, or we call it target-based, that we kind of design work based on different type of data. If they are learning from a cell-based assay screening data, they can basically predict if a molecule is effective or not by just take some cell-related image data or some cell fluorescence data. But if if they know the target 3D structure, the AI can quickly design a molecule that fits that target. And another challenge is synthesis. Actually, in drug discovery, synthesis is the most expensive chain of the industry. For example, any given new chemical, it's about $10,000 of cost to synthesize it. So, and if we want to screen 500, guess how much that costs? About $5 million. So it's just really big cost. And then to find the best synthetic route, especially for natural molecule, it's a huge demand, because that cannot be done by human. But just about 2020, that's 3 years ago already, those type of synthetic planning AI already can plan like natural product synthesis that's people really can't really tell if that's a expert design or AI design. So I do have confidence on AI that it can eventually achieve the automatic synthesis system that really tremendously reduce the cost and the sampling speed of the space. And another thing is like the AlphaFold, which we know is a big event in the field, because the fundamental biology of how protein is fold inherits a lot of knowledge about how it functions, and also how could we use drug to target its function. Like this is one of the simulation I've done earlier in my postdoc year, that is you can say that we just say this is basically two comparison, a protein with a drug and a protein without drug occupied in the middle of the pocket, and you can see the dynamically very different orientation. So even it's the same protein, so which means like not just the 3D structure, the dynamic 3D structure would determine the final biological function. And how could we, from the primary sequence, directly predict what's going to happen at the 4-dimensional 3-dimension plus time event? That's create a huge challenge in the past, but at least right now we'd be able to really predict the 3-dimensional result, and then be able to have some insight. And at the same time, we also find out if you just model the primary sequence using protein language model and directly correlates to some function, it can also accurately predict the function result goes through the folding process. So that's kind of very interesting observation in the AI field. And another thing is like when we do any type of AI construction, we always find out the data is not enough. and there's always limitation of data. And how do we combat that limitation is basically most of the algorithm engineer has to focus. So those are probably a lot of details. I might not go dive into one by one, but basically we have ways of customizing your deep learning model to a specific data set to be able to make it more focused and then to be more accurate. And also the evaluation matrix, when you are optimize your model, what matrix you use against it is also determining the final outcome of that problem you use. Because one AI model can maybe do 5 things measured by 5 matrix. They might not do all 5 things well, but if you choose the good one, maybe they will have the good result. And also there's also different type of the performance upper bound or you can play with, and also how to quantify uncertainty. Since most of the AI models are probabilistic models that you basically the probability is the output. So we play that basically is the uncertainty game. And how do we identify certain uncertainties? There's this uncertainties epistemic uncertainty mostly caused by insufficiency of data, and there's also other toric uncertainty, which is actually how you observe the data. Will cause the uncertainty. So those are the ways. There's different ways to design your algorithm to combat that. And also latent space manipulation is also a very interesting way, which is how we manipulate the black box. So we all know that the latent space is a black box that when the AI learns something, it basically creates this thousands or even billions of dimensions of parameters to describe a problem, instead of human just uses our 3-dimensional or our 5 sense to perceive the world. So in that case, in the latent space, there is a huge room for manipulation, and we can really distill certain information, useful knowledge to serve our needs. And then so basically, there's various ways to combat the data limitation. And then also you can use simulate or semi-simulate some data to if there's a data gap there. So those are kind of technical. And here I would just introduce some of the real industry case that I'm doing in my company. That first of all, the small molecule design. So when we try to design an engine, so normally we have to consider from binding, selectivity, optimization, and how to generate new drug-like molecule, and pharmacology, like all aspects of pharmacology need to be considered, and also off-target screening. Like we have like 20,000 genomes in our human that we only want to target one proteins, and how we avoid attacking all the other proteins and nucleic acid. So all those are the if we're computationally done them, it would be very easy, because it's basically it's just maybe one model you replicate 40 times with 40 different data sets. But for experiments, maybe it's very hard. So that's why we would like to make all those calculation in the front before we actually go synthesize the molecule. So normally this process would filter us from a virtual space of 10 over like a billions of molecule into 10 to 20 candidates. And then we can reduce the number of trial and error in the end. So this is kind of the whole process. We can start with a protein sequence, which is identified target, and then we start to design screening, and then we propose 10 to 100 candidates for both small molecules or large molecule, and then we do the wet lab screening. And we often do multi-objective optimization, and this is all the pharmacology like tests normal the industry wanted to test for any given chemicals, which each one of them is a independent assay has to be carried out, and then this is some of the performance of the AI model, how accurate it is as a mimic to this actual wet lab assay from absorption, distribution, transportation, metabolism, and toxicity, PK/PD, and tissue distribution. And all that has to be considered in when we design a drug. And also for off-target, there's commonly known off-target drug panels that the industry offer for any given chemicals, just trying to avoid, because those are some of them are they are a therapeutic target, but at the same time, they're so important physiological functions that if you hit that, there will be system toxicity. So those are the domain you need, and each of them are an independent AI model as well. This is some of the metrics we compare between the software and the data volume, and one inventions we did is we compare like the average, like a docking software we used in the past, and to use structural sequence-based AI model to design, to encode molecule and proteins, and then to make prediction. We found actually pretty good improvement because the data set is much larger. So in that case, we have some real case. For example, this is like one of the SOS1 inhibitor we're trying to discover for a partner. And then we basically screen like billions of possible scaffolds, and then we take and then the 10 of them we think computationally have the best score, and then we go synthesize them, and then we test them in a group of different assays. This is a biochemical assay, and there's some in vivo assay, and and also the control medicine we use for this from this German company, the BI3406. It has a series of we call the liver toxicity problem caused by the CYP metabolism. So and here we will be able to improve using our new design. So, and this is the whole process is just about 2 months we finished the project, and then all those green ones are positive data that is compared to the clinical candidates we're trying to improve. So this is still ongoing. So this is just like some real case, and then this one is we collaborate with Sun Research Institute. So this is a transcriptome factor which binds to DNA that with a very flexible conformation. It's impossible to use conventional docking to do the screening. So we use our AI engine to do the screening, and then we purchased 20 compound from 23 vendors of the world that we computed successful, supposed to be effective, and there are 7 of them are hits. And then we also using our different type of scoring system and compare different virtual screening methods, and find out that our scoring system is aligned with the actual rank of the experiment. So this is some of the experiment data. And we can also do more complicated PROTAC design, which is we call is a degrader type of larger molecule, and we get also 10 compound, we get the hits. So those are and this is like the interface of the software, and it just inputs some of the design input, and it will generate molecule automatically. And it will give them like comprehensive drug-like score based on different AI model prediction. And we can also do like protein, like antibody design using similar ways. And we benchmark the accuracy and also benchmark the computational cost is dramatically improved. And then we develop an antibody for the COVID in just 2 months. And this antibody is also mutation resistant. When we calculate it, at the time only there's delta strain, we find later this antibody is also effective to the Omicron, and has pretty we tested 50 out of billions we compute, and this is like the hit rate, and then against the different strains, and also some of them against Omicron. So this is, I may not go into the details too much, but basically, I think at this time, compared to like 10 years ago when I do the similar work, I think AI is really improved the accuracy of the computational results compared to experiments like dramatically. So in the past, I would say like 1% hit rate is already the best of the art, but now we can do up to like 40% hit rate. So which is pretty high with the advanced algorithm. So then, yeah, we can also do multi-target affinity maturation, and this is experiment validation. All the light colors are hits from the computation. So, and we can do de novo or antibody design or like antibody affinity maturation in just about 50 proposed candidates. We'll be able to find the candidates. So this is some of the work that the company is doing. And here, when I started the company at 2021, I had this vision of how the AI drug discovery industry would shift. I think from right now to 2030, like most of those different AI model and robotic and would be highly integrated and to impact the industry. So and there's tremendous algorithm advancement. So that basically we were still using the old framework, but every step will have a huge expedition. And I think between 2030 to 2050, the AI would do the next level more, basically on two impact. One is it can plan a research on its own instead of human plan and put the AI in the picture. So basically they can come up with the best plan for how to do this research and managing the entire drug discovery operation. And the second is knowledge reconstruction, because we think right now all the medical informatics and bioinformatics, all those terminologies or terms or observables are defined by human, but they're not the best and they're very redundant. They're not completely the most efficient or elegant way to describe biology. So I think maybe AI would help us to redefine all the terms in biology more elegantly and more mathematically computable. So in that case, we would the noises of the data and then the knowledge system would be much less. And then and biology will be more of a hard science instead of a soft science in the future. So I think that's what it would do. And in the end, I think we can achieve autonomous drug discovery. Like I said, the entire bench to bed to bench precision medical loop by a completely robotic lab, or even we call the lab on the chip, to conduct all the wet lab experiment without human intervention. And at the same time, all the data will also be connected. So the computational system will be completely autonomous as well. And hopefully that can be achieved very in the near future. And to achieve those, we really need to integrate a lot of resources. We need experts in drug discovery and algorithm engineering, robotic engineering, data engineering, and even blockchain engineering for certain data project. So I think basically I just hope this vision is big to be achieved by a single company. So I think it's basically the entire industry and academia and institutes and also companies can work together and to achieve this goal eventually. So this is my talk. Thank you very much. Yeah.